All right, so why do I want to record? Because all lectures, at least all my lectures, I will do a recording, and then I will post a link for this particular lecture on Spectrum, so that you guys can always go back to Spectrum and review the lectures if you need to. Okay, especially if you are going to do some revision, and there are some things that are missing in your lecture notes that you want to review, then you can always go back to Spectrum and then find the corresponding lecture. Okay, so normally I'll put I will name the lecture as the date of the lecture itself, and then I'll put lecture 1, 2, 3, 4, and 5, and so on and so forth. Okay, and the number for the lecture is reflected on the number of week of the lecture itself. So, but for this subject, it will be a bit confusing, but I'll sort it out later on. Okay, but if you just wanna find which lecture it is, just look for the date itself. Okay, for today, 27th of April will be should be lecture number 1. Next Thursday, there won't be any lecture, so there's no confusion for this week. But for next week, I'll try and figure out the best way to actually rename all the lectures. Okay, so the course content, what you will be learning in the next 7 weeks are these two things, okay? Proteins and of course peptides. Why do I like to talk about proteins and peptides is because this is actually my field. My field of research, my field of study is basically proteins and peptides, okay? So we'll be talking a lot about protein and peptides, and then followed by talking about nucleic acids, DNA, RNA, how do we actually do protein biosynthesis from an RNA or from DNA to RNA and then to protein. Okay, so we will look into that. So, yep, I'll just continue. So course content, which you already basically have 4 course contents. Okay, and these 4, how are they actually related to what I'm gonna talk to you is basically number 1. Okay, so what you need to at the end of the semester, what we hope you will get is basically you'll be able to identify structural features of carbohydrates, lipids, proteins, and of course nucleic acid. Okay, nucleic acids. And then number 2, to explain structural differences in nucleic acid. Well, structural differences between, of course, nucleic acids, all these 4, okay, and their roles in protein biosynthesis, which is specifically related to nucleic acids. Okay, number 3, illustrate the use of carbohydrate, lipids, and nucleic acids in industrial processes and forensic. We will talk about it. We will talk about those around week 7, 8, 9, around week 10 or 11. Okay, and then illustrate the synthesis of peptides and the sequence analysis of proteins. So these are the things that you'll be learning. Okay, and I know for the past 7 weeks you have learned about carbohydrates and lipids. Okay, so hopefully that you grasp the concept well, and therefore you'll be able to pass this course with flying colors. Okay, so lecture timetable, we are actually here. You can screenshot this if you want. Okay, so basically next Thursday we will have our Nuzul Quran holiday, and then the week after that we will have asynchronous. Okay, so the importance of this slide is to look at this, either asynchronous or asynchronous. Okay, so asynchronous in my case basically lectures that will be pre-recorded, and then I will post a link on Spectrum, and I hope that you guys can actually use Spectrum to watch the lecture instead of going to the YouTube, because I will upload the lecture to YouTube first, and then I will link it to Spectrum. And then why I want to use Spectrum is I'll talk about it soon. Okay, just hold that thought. I'll just continue with this. Okay, so and then followed by that we have another asynchronous lecture, and then the week after that is the Raya week. So I'm sure this is on Thursday. So it's very good because the Tuesday on the same week, which I believe some of you might, I'm not sure whether we are all be celebrating, but the normal way is for us to celebrate for the whole week, okay, balik kampung and so on and so forth. But because we have a asynchronous lecture, so I can just prepare the lecture, post it on Spectrum, and then you guys can just, look at it whenever you want. Okay, hopefully before the quiz. Okay, because straight after Raya, you will have your mid-sem break, and then the first week, the first Tuesday following the mid-sem break is when you have your first quiz. Okay, first quiz from my section, so it will cover these topics and these topics only. We will not be covering about nucleic acid. Okay, and then the week after the quiz, it depends on how fast I can give the lecture. If I can give out all the materials have already been covered, then we will do a tutorial discussion on the quiz. Otherwise, we will have a lecture. So, and the following last 4 lectures will be on nucleic acids. Okay, so these are all planned lecture slots. I will definitely change here and there, but something that you need to be sure is that that date and that date will be the date whereby we will have your quiz. Okay, so you have your quizzes on those 2 dates. So summative assessment for your final exam, 35% will come from me, 35% will come from Dr. Diniz. And we will have, I'm not sure if she actually mentioned about it, we will have an MCQ short answer question as an online version. Okay, so we will have an online quiz. We will not be using Spectrum. We will set it up using Google Form, so that I'm hoping that everybody will get a good connection. Okay, so at least during the final exam week, it won't be a hitch, and hoping that everybody can submit the answer sheet within the time limitation. Okay, so it will be 2 hours and it will be during exam week. So of course you will have 1 week for your study week. So the following week after this is your study week, and then followed by that you will have your exam weeks as usual. Okay, so lecture plan, week 8, 9 until week 14. So this is the lecture plan. Again, as I mentioned, there might be some changes due to our public holidays. And I'm not sure what the government will do next. If they will impose another MCO, then I might have some difficulty in conducting some of the lectures. It won't be like this, meaning that you might not see me, but you can see the slides, okay, something like that. Because in my office, I have my own PC, so I'm actually having this lecture using my own PC. But if I have to stay at home, then it will be a bit difficult to actually have the same setup, okay? All right, and this is very important for you guys. The lecture won't be a one-way lecture like this, okay? What I normally do, or at least what am I doing for some of the other courses that I'm teaching, is to actually use this option. Okay, so what I want you guys to do is, for all live face-to-face lectures, okay, we will be using this application. It's an app or a website. You can just open your browser and then go to paulf.com and then key in this as the username. So it will be paulf.com/hadifuad049. Okay, and then what you will see there is basically I'll be activating some questions. So as we go on during the lectures, I'll be activating some questions, and then the question will pop up on your screen. Then you can join in and test your knowledge. Okay, and there should be 70 of you. Now it's only 59. So the first 40 students who answer whatever questions that I'll be giving out will have the answers shown to you guys during the lecture itself. Okay, I'm hoping that it will go without a hitch today. We will see. Okay, and additionally, for all pre-recorded lectures, why I ask you guys to watch on Spectrum is because of this. I will use a H5P element whereby I can add in questions during the lecture. So instead of having, if you're having a live lecture like this, I can use poll everywhere. So I can post the question to you guys, and then you guys can join in by answering the questions. But for all pre-recorded lectures, you cannot do that. We cannot do that. Therefore, I'm using this module called H5P on Spectrum. For you guys, it will just be like a YouTube recording. You just click on play, and then at some time points during the lecture, there will be a pause and a question will pop up. Okay, so you can have a test, have a go at your understanding or at whatever questions that I'm going to prepare for you guys. Okay, so basically this is like a Paul F, but it's for pre-recorded lectures. Okay, all right, so get your phone now and QR code this. So this is for today's attendance. We'll post an attendance screenshot like this. So scan it, and then you will need to key in this secret code. Okay, I will change the code every week. Okay, so I will definitely change it. And those who actually have listened to my lectures, then I know that you guys are actually present. Those who missed the lectures, then I know that you guys did not watch either the recording nor the live lecture. Therefore, your attendance will be showed as absent. Okay, so QR code this quickly. Otherwise, I will have another slide on this particular QR code where you can just go in and quickly key in your attendance. Now, so open. You can actually download this PolF application. So it is called as PolF for iOS and Android. Okay, so you can just download this in your mobile phone, or just for today, you can just quickly open a browser, a small browser tab. Okay, key in this very quickly, paulf.com/hadifuad049 on your browser, and we will do our first revision. Oh God, I need to do this. Okay, now just one moment here. Why won't you open? You won't open.
Mine also cannot.
Yeah, I know. Because I need to. I think UM internet is so slow now today. I don't know for what reason, for whatever reason. Oh, there you go. Okay. I need to do this quickly, and then what will happen is once I activate the question, okay, I can activate the question now without having this. So while waiting, look at your phone and try and answer this question. Okay. So this is question number 1. Which of these is the function, should be functional group for an amine? You guys go ahead and answer that quickly while I'm doing this. All right, internet is a bit slow in this afternoon. Yesterday when I tried at home, it was very quick. Let's see, is it working? Is it working or is it not working? Can you guys see the questions now?
No, doctor.
No?
Cannot, doctor.
I mean not on my slide, but if you go to your browser and then you key in polf.com/hadifuad049, you should be able to see it.
Yes,
Yeah,
I
doctor.
can.
good.
Can.
So just fill in that one first.
Doctor,
I
can I
can
get
open the
the
questions.
username?
Hadifuad049. Slides. That one. So the username is this. See, the internet is so slow. It is not able to log me in. Backup plan. I have my backup plan. I don't know what happened. It keeps on asking me to do this. Okay, so backup plan just for these questions. Let me see this one. Present window, share now, and that one. Okay, so this is what you will see next time, hopefully when it's okay. Okay, you will see something like this. Okay, so it's a live poll. So which of these is the functional group for an amine? Okay, 86% says the RNH2, 11% says RNH3, and then 3% says RCOOH. What's the time now? How many? Poll. How many of you have answered? I have no idea. Okay, never mind. Okay, so the correct answer is RNH2 and NH3. Okay, so those two are actually correct. The first two options are actually correct. I just put it in to confuse you guys. As usual, those who actually know me, then they will know that I like to confuse students. But I will let you know the correct answer. So number 1 and number 2 are correct. Okay. All right. So revision number 2. A carboxyl can react with an amine to form an amide spontaneously. True or false? I'll give you guys 1 minute to key it in or choose the answers. True or false? A carboxyl can react with an amine to form an amide spontaneously. 59, 40, 61. It's like a race, you know, it's like a race. So come on. May the best team wins. I would like to see how many responded. Okay. All right. So majority of you says it's false. 38% says it's true. The correct answer is false. Okay. So carboxyl cannot react with amine spontaneously to form an amide, but it can react. So carboxyl can react with amine spontaneously to form salts. Okay, salts. Then the answer is true, but to form an amide, the answer is false. Okay, so it doesn't spontaneously form an amide. If you just make a carboxylic acid and an amine, you put it in, it will take a while for that for them to actually form an amide bond. It might take like a whole week or a whole day. It's not as spontaneous as we expected. It forms salt very, very spontaneously, but not amide. Okay. All right. Last question. Which of these is an amino acid? So you can click on the correct image. Which of these is the amino acid? Is an amino acid. Ini the one at the bottom macam saja je klik ke accidental click, I'm not sure. Okay, one more. Okay, all right. So full. So the correct answer is the top left. Okay, so top left is the basic structure of an amino acid. Top right is the structure of a nucleic acid. Okay, bottom left is the basic structure for a lipid. So one of the structure of lipids. So you do have a lot of structures. This is the one of the basic structure of lipids, and the bottom right is glucose. Okay, so good. All right. So, but because nobody actually because this cohort did not get 100% right to choose the top left hand side, then we need to have a lecture. Okay, I'll now. Okay, so revision 1, revision 2, revision 3, an introduction, lecture 1, structure, classification, properties of an amino acid. Okay. All right, so in the biological, on molecular biology field, there is one central dogma, which was coined by James Watson and Francis Crick back in 1958. So just 1 year after our Independence Day. What does it mean by central dogma? So central dogma means a framework for understanding of a transfer of sequence of information between information carrying. Okay, so this is what central dogma means in general. So what it means for molecular biology is that a basic principle from our genetics, which is DNA. From DNA, it forms an RNA, which is the material that is carrying some part of the information from our genetics. Okay, and then towards the end, the body will synthesize proteins. Okay, so this is the basic ideology way back when DNA was first, the structure of DNA was first discovered by James Watson and Francis Crick. So the presence of DNA was known for quite a while, but the structure of DNA was not identified until about 1958. Okay, so this is a central dogma, meaning that for so long, this is what all biologists or biochemists think as kind of like the Bible or the Quran of molecular biology. So you have our genetics, then from genetics, genetics or DNA. So DNA carries a material in the form of RNA, and then RNA being translated into proteins that makes us human or makes our body being able to do what it needs to do. Okay, so that's the original ideology. However, if I'm not mistaken, around 1980s, okay, that central dogma is no longer an accurate information. So it is still a basic nature on what our body does, meaning that from our DNA it forms RNA, and then from RNA it forms proteins. So that is still correct. However, when we are talking about the flow of information, okay, so central dogma means there's a framework of flow. So A becomes B becomes C, and it cannot go in the reverse. So that's the central dogma. And nowadays it's no longer inaccurate because RNA now can go back and produce DNA. And how do we know this? Because HIV, okay, everybody knows human immunodeficiency virus that cause AIDS. Okay, this virus is very unique in that it has an RNA, but it also has a protein that can reverse transcribe from an RNA to become a DNA, and then this virus managed to put in their DNA into our cell, okay, into our genetic material. Very difficult of the presence of a protein from HIV that can do that and then put in the DNA into our genetic material. Okay. All right. So enough with central dogma. We will talk about RNA in the next coming few weeks. Okay. So for today, we will just focus on peptides and proteins and looking at the basic structure of peptides and proteins. Okay. So that is the basic central dogma. From DNA, you have DNA polymerase. It will replicate DNA sequence into DNA. At the same time, a different proteins called RNA polymerase can transcript. They call it, the process is called transcription. So it can transcript a DNA into RNA, and then from RNA it can be translated in ribosome to produce proteins. Okay, and proteins has a lot of function in our body. So the building block of proteins, as you guys have already known, is the amino acids. Okay, and how it is formed is that all these amino acids are covalently linked to one another, one after another, forming this polypeptide, tetrapeptide here because we have 3 amino acids. So we call it 3 as in 3, okay? And peptide is because it's a peptide. It's no longer an amino acid. So when you have one amino acid reacting with another amino acid, it forms an amide bond, or we can also call this as peptide bond. Okay, so that's why this structure over here, we call it as a tripeptide because it has 3 amino acids instead of having 3 peptide bonds, but it has 2 peptide bonds, but it is called as tripeptide. Confusing, but you know, we are just the younger generation. We just use whatever is being coined by the previous generations. Okay, so that is the basic structure of an amino acid. So the first question that I asked, which one is an amino acid? This can be also considered as an amino acid. Okay, so the basic nomenclature of peptides also follows what chemists normally do. For 2 amino acids, we call it as dipeptide. 3 amino acids, we call it tripeptide. And then when you have a very long polyamino acid, you can also call it as oligopeptide here, or you can also call it as polypeptide. So oligopeptide is normally when you have a nucleic acid, okay, conjugated to a peptide. So we sometimes call it as oligopeptide. Okay. If you have a glucose or a carbohydrate group connected to a peptide or protein, then you call it as glucopeptide. So it's the basic nomenclature for a general term. Of course, it's not the chemistry like an IUPAC name, but this is just a general nomenclature that we use. So definition by the Cambridge Academic Press defines proteins as any of a large group of chemicals that are a necessary part of a cell of a living things. Okay, so that is a definition of a protein. A definition of a peptide is a chemical compound that is made of a small chain of 2 or more amino acids. So we have one amino acid, you don't call it a peptide, you don't call it as monopeptide, you just call it as amino acid. But once it is conjugated to another amino acid, you can call it as a peptide or a dipeptide. Okay, so it is a very gray area between the definition of peptides and proteins, because as you can see here, the definition for peptides is chemical compounds that is made up of 2 or more amino acids. It doesn't talk about function. But when you talk about proteins, then it says that it's a necessary part of a cell of all living things, meaning that it needs to have a function. So that's the kind of like a gray area. So some things you can consider it as a proteins, some you can consider as peptide, but there is no distinguishing barrier between a certain number of amino acid, you can call it as a protein, certain number of amino acid, you call it as a peptide. So there is like no clear distinguished definition. As an example, definition number 1 mention that or define proteins as polypeptides with 40 or more amino acids. Additionally, in a different research article, okay, tau protein in Drosophila is only 11 amino acids. So you can consider a short polypeptide or as a protein as long as it has a function. So it doesn't really involve numbering, number of amino acids, but more on whether it has a function or not. But even then, peptide can also have a function, but you still consider it as a peptide. So it's a very, very gray area. Now, but the takeaway message from the confusion is basically amino acid builds peptides and proteins. So you have one amino acid, it can form peptide, and then as it grows larger, it will form a more complex structure that we normally call as protein. Okay, but there are also peptides that are found in nature, which is still huge, and it has a function, but people still call it as peptide instead of a protein. let's look at the so generally there are 20 naturally occurring amino acid. If you read general articles, if you just Google, you might find that some manuscripts mention there are 21 naturally occurring amino acid. Some might say there's 22, but regardless of the numbers, the International Union of Pure and Applied Chemistry, which is us, okay, only defines 20 naturally occurring amino acids. Okay, so if you count the structure over here, so you have 5 over there, 4 over here, 4 over here, and then another 3, 6, 8 over there, you will have 21. Okay, so this structure has 21 amino acids. So which one is not part of the 20 naturally occurring amino acid? I want to have a guess, but probably we don't have enough time, which is this one. Okay, a selenocysteine is not part of a 20 naturally occurring amino acid that is being recognized by IUPAC, but this figure here that I've got from an internet says that it's part of it, but in reality it's not. And also, sorry here yet. And also, even though we say that there are only 20 naturally occurring amino acids, but this statement is only hold true for humans. Okay, for humans, there are only 20 naturally occurring amino acids. Of course, our body, if you really analyze your body, you can actually find more than amino acids. But the naturally occurring ones, meaning that the same amino acid that we have in our body, if you go outside into the nature, and you analyze, say, a cat or a dog or a tree, then the similarity between our amino acids are only a maximum of 20. Okay, so that's why 20 is the current number of amino acid being recognized by IUPAC. And also, if you look at this figure, okay, so that is a full name of an amino acid. At the same time, you can find a 3-letter code of an amino acid over there. Okay, so there's 3 things. So the full name, the 3-letter code, and of course that one is the single letter code of an amino acid. So in the exam, I might ask you, what is a single letter code E stands for? So you don't need to memorize, of course. This is just a information. Okay, you don't need to memorize. What I will do is I will provide you guys with a list of amino acid and the molecular mass. And if that kind of question actually pops out, you just need to find out. You need to know where you can find the correct information and answer it to be able to get a mark. All right, so an amino acid is a compound which contains an amine group and a carboxyl group. Okay, so that is the basic of an amino acid. So any structure, regardless of what's the length, as long as it has a carboxyl group, a full carboxyl, and an amine group, so you can consider it as an amino acid. But an amine group, a carboxyl, so 1-to-1. If that molecule has, say, 3 carboxyl group and 1 amine group, you don't really consider that as an amino acid. You might call it isomeric amino acid, but it is not an amino acid. Okay, so for this lecture, we will only focusing on the naturally occurring amino acid. So all examples that I will give is all will be all focusing on the naturally occurring amino acid, as per I mentioned in the previous slide. Therefore, the definition can be redefined. So this is my own words. Okay, a compound that contains an amine, NH2 group, and a carboxyl group, and an R group at the alpha carbon position. Okay, so this is the alpha carbon. If you look at the structure of amino acid, the carbon in the middle there, we call it as an alpha carbon. So that is an R group attached to an alpha carbon, and that is the alpha hydrogen. Okay, so alpha carbon is a stereogenic center. Therefore, you know that you can have a 2 mirror image of an amino acid. So you can either have an L amino acid or you can have a D amino acid. So recall, if you still remember, what is an L and D concept in the heterogenic center. Okay, I'm not going to touch about it. If you forgot about it, then go back to your first year lectures and then find information. Okay, so let's take a look at the reactivity of an amino acid just for a short while. Oops. Okay, the first one, at the amine group, you have a nucleophile. Okay, why do you consider it as a nucleophile? Because it likes to donate their electrons. Okay, a nucleophile. And this is also a Bronsted acid, meaning it likes to donate protons. Oh, sorry, it likes to accept protons. Okay. At the same time, on the other side of the carboxyl acid group, you have the hydroxyl of the carboxylic acid, which is the opposite of boracic acid. It's a boracic base, which is a boracic acid. So it likes to donate a proton. Okay. Additionally, at the carbonyl group, you will have a potential electrophile. Okay. So meaning that it can accept electrons from, say, for example, an amine group. Okay, but of course it doesn't do that spontaneously. And finally, you have the alpha hydrogen, which is a weak acid. Okay, so in some instances, you can actually see, well, I'm not going to show you any example, but an L amino acid can be turned into a D amino acid because of this. Okay, so the alpha carbon is a stereogenic center, so that determines whether it's a D or an L amino acid. But you can actually produce a racemate from an enantiomerically pure L or D amino acid. Okay, so it can happen. Therefore, you know that it is a weak acid. And why does it mean by the definition of acid? Is it likes to donate its proton. Okay, so if this alpha hydrogen can live as a living group, okay, you can have an R group there as a planar, and then depending on which direction the hydrogen comes in again, you can either have a D or an L amino acid. All right, so what does this also mean is that amino acid is a very reactive at different pH. Why? Because at a certain pH, you can activate this region. At a different pH, you can activate this region. And at a certain pH, you can either dissociate or cause a racemization to actually occur. All right, and in general, a basic structure of amino acid in a very what do you call it, ideal situation, at pH 10, an amino acid forms a zeta ion. Okay, again, what does it mean by zeta ion? Is basically a molecule that have a ionic and an ionic at the same time on the same molecule. Okay, so you can have that becoming carboxylate, and an amine there can accept proton, and therefore becomes a plus. All right, so about the R group. So the R group is also called as the side chain of an amino acid because there are 20 amino acids, therefore there are 20 different R groups. Okay, for example, as shown here is alanine, which is called as 3-letter code is Ella, single letter code as in just an A, has a methyl group as the R group. Okay, as you go around, you have serine here, you have a hydroxyl functional groups, cysteine, you have a thiol functional groups, tyrosine, you have phenol-like functional groups, and then a tryptophan where you have this bicyclic structure. Okay, so different substituent, so there's no amino acid that has the same R group except for glycine, whereby the R group is actually just a proton, just an H. Okay, so it contains primary amine at the N terminal except for proline, whereby the amino acid The amine group is actually a secondary amine. So most amino acid will have an NH2, but for proline, it only has NH structure because that is the structure of a proline. Okay, so the N has two connecting bonds, and therefore only leave one bonds for hydroxyl. So most amino acid contains iron E in the naming. So alanine, serine, cysteine, tyrosine, except for some. For example, tryptophan, you can have glutamic acid. Okay, so but most of it has an iron E in the naming. Each amino acid can be written as 3-letter code, single letter code, or a full name. Okay, so you cannot click here. If you click here, you go to wiki. So this is just an example of all amino acid, all the structures. Okay, the full name, the 3-letter code, the pKa, and so on and so forth, and of course the pi, or the state, or when pH equals to pi, you will get the Z2-ionic of the particular amino acid. So if you look at here, this are in the real situation is no longer an ideal situation. So you will not be able to find an amino acid which has a pi of exactly 7. Okay, so this is just an just to show you different amino acid, different functional groups that it has, different R group that it has, and of course a different nature or different groups that you can actually bring them together. Okay, so you can have a polar side chain, for example, in aspartic acid and glutamic acid. You can have serine, which also has a polar uncharged. Okay, so this one is polar charge. You can have polar uncharged residues such as asparagine. Okay, so that's the basic structure. So these are all the R group. So do not get yourself confused. So these are just example of the R group. You have serine, glutamine. You have threonine. Okay. Similarly, you have another group which is non-polar and hydrophobic side chain. Okay, you have alanine, which is just methyl, phenylalanine, which has a phenyl group, isoleucine, leucine, methionine, valine, and tryptophan and tyrosine. Okay, so these are just some examples of amino acids, R groups of an amino acids. So the amino acid can also be grouped differently. So some people wants to group the amino acid differently. It's fine. There's no right or wrong, because towards the end of the day, you just look at the structure of the amino acid and then you can group them together. Okay, so you can have heterocyclic amino acid, for example. So there are 3 amino acids that belong to this particular category. You have a benzene containing amino acid, which is tyrosine is one of them, or phenylalanine. Okay, hydroxyl contain also tyrosine and can be serine. Sulfur contain, you have 2, cysteine and methionine, and you can also have aliphatic, which is alanine, and so on and so forth. Okay, so this is just a test. There's no need for you to do it, but if you just want to play around, identify what are these. Okay, so just to test your understanding about the R group and the categories that you can actually bring the R group in. So how are amino acids are made? So in some books in the literature, proteins exist in our body, and they are made up of a combination of 20 naturally occurring amino acids. Again, based on IPAC system, it is actually correct, but these statements only refer to 20 L amino acids. So if you consider the presence of D amino acid, this is no longer a correct fact. Okay, why? Because there are some microbials, so living things. So this is not just something that you can make chemically. So living things, a bacteria forms or construct their cell wall using a D amino acid. Okay, and some bacteria also uses or synthesize their own D amino acid and then use it as an antibacterial agent, such as a Bacillus subtilis from a Tracy strain. Okay, so some of the strain does not produce a D amino acid, but in this particular case, it actually produced a D amino acid. And if you don't really remember what does it mean between a stereogenic center, mirror image, and so on and so forth. I have a picture over here. On the right hand side, you have a D isomer of an amino acid, and on the left hand side, you have the L isomer of the amino acid. Okay, so combination of acid results in a protein of a different characteristics and function, which we will look into. So if you have all 20 amino acid, it will function in a certain way. If you have all the amino acid, it will function in a certain way. If you have a mix of them, it will also behave differently, and it can also have a different characteristics in the sense that proteins or short peptides might not be as stable, thermally stable, or proteolytic stable. So meaning that it's difficult for them to dissociate into individual amino acid. But if you actually mix D and L amino acid, they are more stable. That is one example of a different characteristics of what a combination of amino acid can do. So in human in general, there are 10 essential amino acid and 10 non-essential amino acid. So what does it mean by essential amino acid? This is basically an amino acid that our body cannot produce on our own. So the only way where we can acquire this 10 amino acid is by eating. So the more you eat, the more amino acid that your body can reuse and build up the protein inside your body. So that's why you can see that in Africa or in places whereby malnutrition is part of a concern. Okay, so you can see that the body is very weak, even though they do eat, say, banana a lot, or they eat a lot of potatoes. So they can acquire a lot of energy, so they can still survive, but the body is very weak in the sense that there's no muscles and so on and so forth. Okay, so that is the definition of essential. And the opposite is true for non-essential amino acid, whereby you can just eat proteins and then or even glucose, and then our body can actually change it from a glucose or from one type of amino acid to another type of amino acid. Okay, so source can also be from an amino acid or even from our food. All right, so this is just the list of the essential and non-essential amino acid. So if you read in the literature, you might see that some people say there's a different number. It's not 10, it might be 9 or might be 11 essential amino acids. So it can varies a lot, because originally back in when people try to group this amino acid, so they grouped it based on function, meaning that this amino acid is required for our body to survive. So they call it as essential. And the rest are the amino acid that the body can produce. So we don't require them from our dietary, but you know, we can still produce it. And if you look at all these 20 amino acid, one is written as a question mark. So it is for you guys to actually fill it in. Okay, so I put in a hint there that it contains a sulfur group. Okay, so can you identify all of these amino acid from its single letter code? Again, it's just a rhetorical question. You don't have to do it, but if you want to do it, by all means, you can actually do it. This is something that you just need to know. You don't need to memorize. This is just a process by which our body convert glucose into different types of amino acid. You can see there histidine, serine, alanine, valine, isoleucine, leucine, and so on and so forth. So these are some of the processes that our body can do from just eating of glucose. One thing that you need to remember, that these processes are not in human alone. So some processes are actually not available in human. Say, for example, if our body do not have this, but we do have it. Sorry, if our body do not have this, we do have that, definitely. Otherwise, we will all be dead. We don't have that. Okay, so the only thing that we cannot produce is just histidine and serine. Therefore, we need to get this from eating a lot of food, a lot of protein that contains histidine and serine. Okay, Similarly, this process continues to the next slide, which I cannot change anymore because it's already time. Okay, so we will stop our lecture here just for today. What I'm hoping you guys to do is, if you have not clock in, so that you can QR scan that one to sign in for your attendance. Oh, sorry, that one is for attendance. The one at the top here, if you can QR code, so I will post this each lecture. So I want to see your feedback in the sense that whether this teaching style is the one that you like, or you just prefer, I'll just babble and no questions, no stopping. I will just go and boom, 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 and then done. Or do you prefer something that we did just now using poll F? If you do like them, then just QR code this and then choose in type of intervention is A, because it's a synchronous lecture. And then in the future, when we have a recorded lecture, it will be a B, intervention type B, whereby I will post questions during the pre-recorded lecture. So if you like the lecture system, by all means, do let me know in the survey. Okay, and any improvement is also welcome. You can just put it there. All right, so there's only like one slide, which is a summary. So we will just cover this in the next week lecture. And for that, I'll end the lecture. Thank you, and have a good break on Thursday. Okay. Thanks, everybody.
Thank you, Doctor.
Thank you,
Thank you so
Doctor.
much, Doctor.
Thank you so much, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.
Welcome.
Thank you, Doctor.
Thank you, Doctor.